Okay. Lab 4 is lymphatic system and immunity. And this is the last lab before your first practical exam. Luckily, this is a rather, at this point in the game, it's a rather brief um, lab. There are, a good handful of structures you need to identify, but they're not really terribly complicated. And we're going to go through not only the notes that I put up there, which actually have a good number of pictures that I used, and then we're going to go through the images as well. So the first thing we're doing with the lymphatic system is just identifying anatomy, identify and describe functions, of each of the structures on the models. And so we're going to be looking at lymphatic capillary, a lacteal in the intestines, lymphatic vessels with nodes, the right lymphatic duct, the thoracic duct, the cisterna tonsils, spleen, Peyer's patches in the appendix, which are examples of the molts, the mucosa-associated lymphoid tissue. We're going to look at a slide of the lymph node, and we're going to look, I think there's a model of red bone marrow. If I don't have a picture of it, I probably won't ask it, but I think there's a picture of red bone marrow on the images of the course page, because that's where all blood cells come from and where B lymphocytes become immunocompetent. Then we're going to look at the thymus gland. What hormone does it produce? What is the function of that hormone? We're going to look at a slide of a lymph node. I believe some of the labs will have you look at the spleen also. I just have you look at one lymph node so you can see sort of the organization of it. You just have to know what area is the cortex, what is the medulla. And then we're going to look at some of the physiology, the immune responses in innate or non-specific immunity and adaptive immunity. So all these things you should be able to identify and describe immunological function for each structure. So surface barriers, like the first line of defense, surface barriers versus your internal defenses, which are all non-specific, and then your adaptive immunity. So I have notes that I think are good examples, or will identify the functions of all these, because they're rather basic functions, but especially at the beginning when we're looking at the anatomy, there's sort of like arteries to arterioles to veins. You're capturing fluid and you're transporting it from one place to another, to another. So the notes kind of go through all this. Kind of from beginning to end, functions of the lymphatic system. And then perhaps we'll come back and see if we did not address any of these things, but you just have to know some of the basics. For instance, surface barriers. We're going to look at a skin model, and go, okay, that is first line of defense. Then mucous membranes, because that's part of your surface line of defense, especially along the respiratory and digestive systems. That we have sweat glands, oil glands, saliva, tears, and gastric juice. They're all part of, this physical and chemical first line of defense. And then we have internal defenses, inflammation. We're going to look at one particular white blood cell associated with that, a basophil, but also fever. We have phagocytosis. What are the two, leukocytes associated with that? Neutrophils and monocytes. And antimicrobial proteins, interferon, complement system, and transferrin. The notes, we're going to go through that. We'll identify functions. That would have to be more of a word question, but I could, and we'll identify this when we look at the other images and things, is that it's the basophil, associated with inflammation, and neutrophils and monocytes are associated with phagocytosis. And then we'll look at T lymphocytes and B lymphocytes. Remember, B lymphocytes are part of the humoral immunity. T lymphocytes part of the cell-mediated immunity. B cells become plasma cells that produce antibodies. T cells produce cytolytic enzymes that are the kind of the physical, have to come in contact. That T cell has to come in contact with that organism. and the hormone that stimulates its maturation is thymosin. Right, back up here at the top, I think the only other fill in the blanks, the thymus gland. What hormone does it produce? Thymosin. And what is the function of that hormone? T lymphocyte maturation or immunocompetence, and that occurs in the thymus gland. Okay, so let me change my share to the notes, and we're going to kind of go through those notes, and then we're going to come back and go through the images, the additional images. Okay, so let me put this in slideshow mode. Makes it easier. Okay, so lymphatic system functions. Generally, it drains and collects excess fluid from the interstitial spaces, that we have lost at the capillary bed. We go in under hydrostatic pressure on the arterial side of a capillary, and that allows water and all kinds of molecules that can fit between the walls of the squamous cells, because a capillary is a single layer, simple squamous epithelium that it pushes those molecules out into the interstitial spaces. And as blood is transported from the arterial to the venous side, now the venous blood here acts as like a hypertonic solution and has an osmotic pressure to pull water back into the blood vessel. And then the venous side of the capillary becomes venules and veins and so forth. Well, the remaining fluid that did not get recaptured in the venous side of the capillary bed needs to be captured and circulated. So we have the lymphatic capillaries, and they act as a kind of an open tube with negative pressure in there, so it sucks up that fluid and any other microbes in the environment, and helps to restore blood volume as well as blood pressure by capturing that lymph fluid and any microbes in the environment. It forces that lymph, and lymph is the fluid we call that's now traveling within the lymphatic system, will pass through different lymph nodes that contain B cells, T cells, and macrophages, and monitor its contents, the contents of the lymph. In the small intestine, we have lacteals, which are lymphatic capillaries that are able to absorb or transport absorbed dietary fats from the digestive system and transport it. And then it also acts to return some of the plasma proteins to blood. But we don't usually use lots of them anymore. All right, so it consists of capillaries to vessels, lymphatic capillaries to lymphatic vessels, to lymphatic ducts, and then they transport the fluid into the right or left subclavian vein. All right, so this is one of those models, excellent, for identifying a tonsil. We started adding the tonsils and using that head picture in the last couple of years. So wherever we have orifices of the body, openings of the body where material can get in or get out, we need to line that with mucus and maybe saliva that has acid secretions, tears that actually have some acid secretions. And where we have entrance points into the body, we need to have some kind of a lymphatic organ to monitor, in this case, the contents of air breathing in and out, where it's entering in the nose and through the nasal cavities, or food that's entering, in the level of the tongue, in the throat. So F in the back there, that's a pharyngeal tonsil, one of your adenoids. Right? It functions as the first line of defense against ingested or inhaled foreign pathogens. Right? Ingested or inhaled, eaten or breathed in. That's its job. It's going to initiate your first line of defense. Or actually, it is part of your first line of defense and helps initiate your immune response. All right, in the small intestine, this is a model of the small intestine. We have D, the green structure there in that villi. Those little finger-like extensions upward are called villi. And D is a lacteal, and it functions to absorb and transport dietary fats. They will be in the form of what's called a chylomicron. These fats are put into the form of chylomicrons, small molecules that are transported then to the liver. As fats are absorbed across the what's called simple columnar epithelial tissue layer of the mucosa, you have a single layer of simple columnar cells. And as some of these fats are absorbed, in their building block form of the lipids, the smooth endoplasmic reticulum of that organ, of those cells, take those fats and they put them in the form of a chylomicron. And then it can be exocytotically released on the other side of that columnar cell and enter into that lacteal. It's an open end tube, similar to a capillary tube or straw, collects fluid from the interstitial spaces. This, similar to veins structurally, as they have valves, but thinner walls. And so we're talking about the lymphatic capillaries. All right, we have the right lymphatic duct and the left lymphatic duct. In lecture, we talked about, and hopefully your lecture class talked about the unequal distribution, so to speak, of lymphatic fluid. And that's not necessarily an unequal distribution, but the way it's transported back to the subclavian veins, it's not an equal distribution. For instance, the right lymphatic duct is not transporting lymphatic fluid from the right leg, the right abdomen, the right arm, the right side of the head to the right side, and the left leg, left arm, left side of the head to the thoracic duct. The right lymphatic duct is transporting lymphatic fluid from a much smaller, comparatively, area of the body. It transports lymphatic fluid from the right arm, the right side of the head, and essentially the right chest. And the fluid from those major regions that will be transported from lymphatic capillaries to lymphatic vessels, ultimately to the right lymphatic duct. So the duct is kind of like, an inferior vena cava, the largest kind of tube system where other, vessels has all merged together to finally use this one point of articulation, to another large vessel. So capillaries merge to become larger vessels that ultimately become two major ducts. You have the right lymphatic duct and the thoracic duct. The right lymphatic duct, identifying B over here in white, is a very short length. Has a very short length and empties into the right subclavian vein. So that blue vessel that it's connecting to is the right subclavian vein. It drains from the right side of the head, the right arm, the right chest. The orangey nodules on this model are lymph nodes. There's a little lymph nodes. The thoracic duct assumes the name thoracic duct when this duct originates just superior to the cisterna chyli, in the abdomen. So the cisterna chyli is a collecting area behind the stomach that narrows down to become the thoracic duct, that white tube kind of, the white tube A, that's the thoracic duct, that you can see here in its entirety. You follow it all the way around the superiorly a hairpin turn, and then it's going to connect, and articulate with the left subclavian vein. But if you go inferior where the letter A is, and you follow it down as it parallels the thoracic aorta, that whole stretch there, that also is the thoracic, is the thoracic duct. And it has that name in the abdomen as well. I'll show you another picture where it backs up on this model, and you can see where the cisterna chyli is. And then from that point upward, superiorly, it assumes the name of thoracic duct. The thoracic duct empties into this left subclavian vein. So if you follow the A and you go superiorly up and around the little hairpin turn, and you can see how it connects to the left subclavian vein. It drains the lymph from all the other areas of the body, other than the right side of the head, the right arm, and the right thoracic area, and the right chest area. So it's the right leg, right abdomen, left leg, left abdomen, left side of the chest, left arm, left side of the head. Okay, this is that same model, but we backed up a little bit so you get a little better overview of what you're looking at here in the abdomen. So C, is pointing to the hepatic portal vein. Behind that is this dilated region where multiple lymphatic vessels are merging. One's draining from the lumbar area and the one's transporting lymph from every place else in your body. From the abdomen, from the two, lower extremities. And they're all going to kind of merge together like two big highways merging together. And it needs to initially dilate before, as the fluid is moving superiorly, from C up to B. And then you can see how B is the thoracic duct. It continues, as the picture prior to this, to make its little hairpin turn and then, connect to or articulate with the left subclavian vein. Dilated, it's a dilated white node in the lower abdomen behind the hepatic portal vein here. Where these lymph vessels all merge. Superior to that vessel, it assumes the name of the thoracic duct. Some people refer to it as a left, a lymphatic duct, but most people are still going to refer to it as the thoracic duct because it's a huge, long duct that transports lymph fluid through the thoracic duct. E is pointing to the thoracic aorta. Okay, so going back to that model from the small intestine, sometimes referred to as the cake model because there's lymphatic, tissue in the walls of the ileum at B. So it kind of looks like a called the ice cream cake. But in any event, B, so D was your lacteal. That's that lymphatic capillary, open-ended, that's going to transport a lot of the fats that are being absorbed from the small intestine. They travel through those columnar cells, and the smooth endoplasmic reticulum forms chylomicrons, and then it exocytotically releases those molecules on the other side, the basement side of that cell, and it's able to be kind of captured and sucked into D, the lacteal. Then that transports that material, the lymph. It's also capturing in the interstitial space. And it becomes filtered by B, which is the Peyer's patch. All right, so we have the lacteal in D, and then we have that Peyer's patch in B. So it filters the lymph, and helps, again, identify pathogens that might be present and initiate an immune response. So all these, these specialized lymphatic structures, part of that molt, are designed to detect the presence of a microbe and then let your body know that there's some kind of microorganism there. And that's what we call initiating the immune system. The spleen, here's a good model that has the pancreas, the small intestine, we have our two kidneys, and then the spleen. Right? The spleen is C. The spleen not only filters blood and has a similar function that a lymph node does, that you're going to monitor for microbes and initiate an immune response. You're going to have T lymphocytes, B lymphocytes, and macrophages. But it also filters blood to remove the old and damaged, red blood cells that need to be removed from circulation. It's the largest lymph organ in the body. It's also a source of red blood cells in the fetus. Produces, we initially start by the precursors, and some of those red blood cells are coming from the yolk sac, but then the spleen in the fetus starts to produce red blood cells. The thymus gland is a gland anterior and superior to the heart. Here is what I call a baby on a plate model, naked baby on a plate model. And B is the thymus gland. A is the thyroid gland, but B is the thymus gland. Okay. And the thymus gland produces the hormone thymosin, and that stimulates T lymphocyte maturation or T lymphocyte immunocompetence. Lymph nodes. Lymph nodes have afferent and efferent, I should say, vessels that transport the lymph fluid that's been captured at a capillary level, from one area of the body to the other. And as the lymph, let's say from your hands or your feet or your legs, starts being transported ultimately to the lymphatic duct, I mean, all the lymph in your fluid in your body ultimately has to be transported to either pass into the right or left subclavian vein via the right lymphatic duct or the thoracic duct. All right. So lymph all along the way, that is going to, from, let's say, from your arms, the lymph in your hands is going to enter into the lymph capillaries, and then lymph will form lymphatic vessels and start traveling toward your shoulder and then to your right subclavian vein. But as the lymph in these vessels, travels toward your elbow, let's say from your hand to your elbow, is that the lymph from your forearm is also entering into the same system and kind of jumping along, just like the way, smaller veins ultimately merge to form larger veins, and they keep merging with other veins all along the way until, they form the two brachiocephalic veins. So the lymph that has now passed through a lymph node, let's say from the forearms, passed through a lymph node in the supratrochlear area, the elbow, or bend of the elbow area, is passing through another lymph vessel that is now merging and transporting lymph from a capillary from, let's say, your bicep muscle area. Well, all that lymph, even though some of that lymph was previously filtered by a lymph node in the elbow area, now it all needs to pass through another lymph node because it gained some lymph fluid from an area that has not been yet filtered. And so a lot of this lymph is going to pass from lymph node to lymph node to lymph node and maybe filtered multiple times before it reaches those subclavian veins. All right, so it's not uncommon for a vessel to enter one lymph node and exit another, continue on and merge with some other vessels and enter another lymph node and exit it, so forth and so on. So the terms afferent and efferent lymph vessels are just telling you which direction the lymph is flowing, if you're looking at a picture. All right, so the afferent vessel is entering the lymph node. That lymph is going to spread out throughout this whole lymph node, kind of like water spreading out in a sponge. And then it's going to recollect, like you're squeezing out a sponge, into one major lymphatic vessel that leaves that lymph node as the efferent vessel. So you have the afferent vessel and the efferent vessels that allow lymph to be transported from one lymph node to another. So entering or transporting to or into the lymph node would be the afferent vessel, and transporting lymph away from a lymph node would be an efferent vessel. Nodes are divided also regionally into a cortex and a medulla. The cortex is the outer portion, the shell. The medulla is the inner region. The nodes have both B cells, T cells, and macrophages there. I don't care which ones. I'm not going to ask you which are more abundant in the cortex versus the medulla. Just know that it contains three types of white blood cells: B lymphocytes, T lymphocytes, and macrophages. And its function is to filter the lymph and activate the immune system. Here is a slide of the lymph node. You can see what looks like rather homogeneous tissue to the outer region, that is the cortex, and B in the inner region. You see how it kind of looks kind of Swiss cheese kind of appearance there? That would be the medulla. And in the cortex, we have these nodules there. We have a germinal center here, where A is, it takes on a lightly staining appearance, but that whole outer region toward the surface is the cortex, and B is the medulla, right? So A is a germinal center of a particular nodule, one little nodule portion of a whole lymph node, right? It's found in the cortex. Right? B is the medulla. you will find B lymphocytes, T lymphocytes, and macrophages, period. So identify, this organ is a lymph node. Identify the region A, cortex. Identify region B, medulla. Identify the cells you would expect to find in region A or region B. T lymphocytes, B lymphocytes, macrophages, or B cells, T cells, and macrophages. All right. And this just is a magnified picture of that cortex region. And it just kind of shows you the difference of the most, like the outer region to a nodule versus the paler staining germinal center. That's more of an FYI. Lymph backslash, spleen slide. I don't use the spleen slide anymore. So, generally the kind of questions would be identify the structure or organ. If you don't see any afferent vessels, entering and exiting, then it's a spleen. But if there are afferent vessels, it's a lymph node. I wouldn't worry about this. I'm only going to ask you the lymph node, and that's all. What white blood cells would you associate with it? T cells, B cells, macrophages. And what would you expect this structure to filter? So let's say I pointed to a model, and I pointed to a model of the spleen. I could point to that model and say, identify that organ, and you would say a spleen. What if I said, identify what cells I would find there? T lymphocytes, B lymphocytes, or T cells, B cells, and macrophages. What fluid does it filter? The spleen filters blood. You can see the splenic artery and splenic vein. But if I pointed to a lymph node, then you'd have different answers, at least to some of those questions. Identify that structure, it's a lymph node. I'm going to ask you the region. What is, I'm pointing to the outer region. A. Identify that region. That's the cortex. Or I could point to B. Point to B on this one. Identify that region. Medulla. What cells might I find at A or B? T lymphocytes, B lymphocytes, macrophages. Something along those lines. What fluid is filtered by that lymph, that lymph node, by this organ? Lymph. Lymph nodes filter lymph. The spleen filters blood. Okay, so now we're moving on to non-specific and specific lines of defense. This is otherwise referred to as the innate. I still refer to it as non-specific, so I need to get out of that habit. But for me, it's easier to keep it straight. Always had, always will. This type of defense does not differentiate between the type of organism it's being exposed to or responding to. It will try to destroy it or die trying. So, these are mechanisms like phagocytosis and fever, that are designed to try to keep working until you either kill that bacteria or virus, you eradicate it, or you just keep elevating your blood, your body temperature, until you finally just kill, denature all your proteins and essentially kill yourself. But that's one reason why you need to watch the fever in patients, and make sure they get iced down, or you modify their body temperature, because that's your body's doing what it's supposed to do. But now that we have more pharmacological applications for different organisms and things, is that we don't want the body temperature to go so high that you denature, the different proteins and hemoglobin and things like that in the body, because you need those to live. So you suppress the body temperature until you can find, what particular drug might work best to try to combat that organism. The non-specific defenses are essentially all the white blood cells except for the lymphocytes. Neutrophils, basophils, eosinophils, and monocytes, or macrophages. The ones that are going to be specifically phagocytic will be your neutrophils and your monocytes. Okay, so your first line of defense, are your skin and mucous membranes. Remember that the skin, has oil glands, so it produces oil, and you have sweat glands, so it produces perspiration. Those secretions, have a rather acidic pH associated with them, so that it acts as a good antimicrobial. It moisturizes the skin so you don't develop essentially cracks in skin and a route of infection should bacteria and viruses be on the surface of your body. And you know they are. When you take microbiology, you're going to learn all the normal bacteria, that occupy your surface, the surface of your body, and then you'll find out, what the abnormal ones that will often, not often, but may colonize, the surface of your body. The fact that you exfoliate, remember that your stratified squamous epithelium of your epidermis, the high mitotic division comes from the stratum basale, the most inferior point of the epidermis where it meets the dermis. You have that undulating ridge, where we said we have cells there that function high mitotic division because they replicate at that lowest level and they push all the other layers of cells upward. And as that happens, you can get dry skin, flaky skin. Well, that's normal. I mean, it might not be normal, sometimes to exfoliate quite so much, but those cells that are the farthest away and toward the surface of the body are going to die off and essentially peel off or exfoliate. And when they do, they take the bacteria that are sitting on top of them. So the fact that you exfoliate, and your body sloughs off the cells on the surface means that it, in doing so, it also takes the bacteria, that is sitting on that particular cell. Now, the bacteria generally repopulate that new area very, very quickly. But it is still a method of, first line of defense is to exfoliate and therefore take whatever bacteria is occupying space on that particular squamous cell. Your second line of defense, are the kind of general non-specific responses once an organism gets inside. So we have phagocytosis, ingestion of pathogens by neutrophils, macrophages. Oh yeah, and natural killers. I always, because they're a small class of cells, I always forget to mention those. Remember, natural killers, are a type of lymphocyte, and they're good at phagocytizing cancer cells, as well as some viruses. We have antimicrobial proteins, transferrin, interferon, fall in this category. We do have complement proteins in this family as well. And again, that's listed in the lab exercises. But the ones I generally ask you about are transferrin and interferon. Transferrin binds with available iron in blood and removes it from being available to bacteria because bacteria like to use iron as well. So you inhibit bacterial growth, by reducing the available iron in blood. And interferon inhibits viral replication. Okay, fever. Temperature too high for bacteria to live. So what we're saying is that there are different chemicals that are going to be released, when we find some of this bacteria, and viruses. And that signals your brain, your hypothalamus, to increase your body temperature because there's some organism there. And, many of these bacteria and viruses are susceptible. They have particular pH ranges they like to exist in, that are the most favorable, and temperatures. Well, a lot of them, you're going to denature their proteins as well if you increase body temperature. The problem is that they're very resistant to temperature changes and they're not really affected. You're going to keep elevating your body temperature, and you're essentially going to start killing off your own cells. So that's the problem with severely extraordinary fever in an individual. But in elevating it moderately, you may be able to kill off certain bacteria and things. Elevated body temperature also increases the activity of interferon and its effectiveness. And the body will hoard zinc and iron to try to stop bacterial growth by using things like transferrin. So the question is, what effect does temperature have on proteins? Remember that proteins, those hydrogen bonds that maintain the secondary, tertiary, and quaternary structures of a protein, are very susceptible to denaturation, which means you break the hydrogen bonds and these proteins just start to unravel, which means they're not going to function the same way. And the two major variables to denaturation are increased body temperature and extremes of pH, especially acidic environments. Okay, then we have inflammation, and the four Uh, Characteristics, of inflammation are pain, swelling, heat, and redness. Basophils and mast cell is a type of basophil. Um, are associated with inflammation because remember, basophils release histamine and heparin, and so they help increase the inflammatory process. All right, then we have specific immunity or acquired immunity. You have your B cells, or B lymphocytes, and T cells and T lymphocytes. B cells found in bone marrow. Matter of fact, all these cells, all blood cells, originate in bone marrow. Right? They differentiate into plasma cells that produce the antibodies. Each B cell codes for one specific pathogen, or one specific antibody, and will leave memory cells behind. So what that means is that once, B cells are, stimulated to become immunocompetent, they will become a plasma cell that makes one specific antibody, that is designed to fit with one specific antigen or pathogen. T cells are targeted by the hormone thymosin from the thymus gland. That stimulates their maturation or immunocompetence. They're responsible for antigen recognition, especially the T4s. T cells migrate from the bone marrow to the thymus to mature. We have three major types. We have the helper T cells, which receive, that communicate with the antigen presenting cells. Then the helper cells stimulate and take that information to the cytotoxics, or the T lymphocytes become specific cytotoxic cells, killer cells, forming specific granules to that antigen that was produced to the helper T cell. And the helper T cell also takes that information, or shares it with the B cells so that now it can differentiate into a plasma cell and make the specific antibodies. And again, whatever cells are left over essentially are your memory cells. Cytotoxic killer T cells kill through cytolysis. So the granules that it produces, need to come in physical contact, with the organism to punch holes in the membranes. And it causes that cell to either lose its cytoplasm or, suffer in some way that it can't survive and it dies. Okay, so that's the end of the notes. We looked at the, let me stop my share. We looked at the form itself, hold on, let me just close out this PowerPoint. Okay. We looked at the lab form that you're going to fill out, the exercises that you're going to fill out for this lab. So now you can use the notes and essentially jot in some of the answers there. Let me switch it back again so we can just kind of quickly go over that. All right, so part one was identifying a lot of the anatomy. We looked at a lymphatic capillary. The example we really looked at was that lacteal, that green structure in the intestine. We had a couple different models that showed lymphatic vessels with lymph nodes. Not so hard. A lot of those were found on the same model that indicated the right lymphatic duct and the thoracic duct, and where the cisterna chyli would be located behind the hepatic portal vein. We looked at a tonsil. That was F. That was that first image and model. It was what I call the half head model. We looked at the spleen. We looked at Peyer's patches. The appendix we're actually going to look at in the digestion lab. So unless possibly we've added now a picture of that appendix, in the other images, which we're going to go through. But generally, we'll look at it on the digestion model, one of those big plastic cookie sheet type models in the digestive system. Lymph nodes, again, found on one of the same. We can look at a slide of it, and it was also on that model where we looked at the right lymphatic ducts and thoracic ducts. And then I believe there should be example in those images of red bone marrow. All right, and we looked at the thymus gland on the naked baby on a plate model. The hormone is thymosin, stimulates T cell maturation. We looked at the lymphatic, lymph node slide, cortex versus medulla. What cells would I find there? And then we talked and we looked at notes about the immune response, the surface barriers of the skin, and mucous membranes, because those are found in the digestive and respiratory systems, as well as parts of the reproductive systems too. That in skin we have sweat glands and oil glands. We produce saliva in the mouth, tears in the eyes. We have hydrochloric acid released from the stomach, that kill organisms. Right? Those are like all first line of defense. And then we have internal defenses, inflammation. So you should know, what are the 4 features of inflammation, and which hormone, exaggerated or increased inflammation, and that was the basophil. So we have inflammation and fever. Fever, increased body temperature to denature proteins in other organisms. It also helps stimulate the actions of and accentuate, interferon. Phagocytosis. The 2 phagocytic cells are neutrophils and monocytes, which are macrophages that leave the bloodstream, or are called macrophages once they leave the bloodstream. Antimicrobial proteins. Here's our interferon, our complement system, and transferrin. Interferon inhibits viral replication. Transferrin inhibits bacterial replication and growth because it binds with available iron that's in blood plasma. And the complement system is a system of proteins that are made that help stimulate this whole process of being chemicals that can help increase body temperature, use chemotactically to find the different bacteria. So white blood cells know chemotactically follow the chemical scent to where they're originating from. And then we have our adaptive immunity, the B cells, essentially the activity of the B lymphocytes versus activity of the T lymphocytes, and know how they work. Know your T4s versus your T8s, and your memory cells. Also in the family of lymphocytes, we said are your natural killer cells, but they're actually part of the, non-specific innate immunity. And then we have our humoral immunity. Those are B lymphocytes that once stimulated, by and given stimulated by the helper T cells that have, the antigen from the antigen presenting cells, they become plasma cells that now start manufacturing antibodies to bind to a particular antigen. So I think reading through the notes too, just kind of over a couple times and then maybe jotting some things here next to these particular terms is probably the easiest way to commit this to memory. So let me unshare that, and then I'm going to actually go into the images now. So let me go into my course content, and we'll go through, I think there are about 10 or 11 slides, or I mean images, from this chapter. So let's click on the images. We'll go to the first one, and change my share real quick. Here we go. Okay, so here we have the first model. This is from the small intestine. All right, so here we have one in green is the lacteal, and then we have the Peyer's patch. Okay, so we have the lacteal and the Peyer's patch. Functionally, the lacteal transports absorbed fat from the small intestine, where we absorbed it in the small intestine. So transport dietary fat, that's fine. And then the Peyer's patch essentially filters that lymph fluid and initiates an immune response from the digestive system, or immune response in the small intestine. So the Peyer's patches are kind of like lymph nodes of the small intestine. So if bacteria gets across that route, then you can initiate a response. Okay, here's what we call the Madonna model. But this is that model that we were looking at the different right and left thoracic ducts. And this, is a little more clear picture than what we had. But one is pointing to all those white vessels. Those are your lymphatic vessels. The smallest ones at the ends would be lymphatic capillaries. Let's see, they all merge at least on the right side to form the right lymphatic duct, and that connects to and articulates, so it transports lymph fluid into the right subclavian vein. 4 is pointing to lymph nodes, little kind of orangey yellow nodules, and 2 is pointing to that white vessel that originates in the lower in the abdomen, behind the pedic portal vein, travels all the way through the thoracic cavity as the thoracic duct or left lymphatic duct, but most people still refer to it as a thoracic duct, and it transports emerges with other lymphatic vessels from the left side of the head, the left arm, the left chest, and then transports that fluid into the left subclavian vein. All right, so we have a lymph collecting vessel, just a lymphatic vessel. We have the left or thoracic duct. I would call it a thoracic duct. I think I use the thoracic duct as a term actually on quizzes and exams. The right lymphatic duct and lymph nodes. This is a good picture of that cisterna chyli. So the other one, you couldn't see it too well because 2 is the pedic portal vein, is actually anterior to it. But if you could peel back some of the anatomy in that model, then you would see 1. That's that dilated region where multiple lymphatic vessels are emerging from the lower extremities, the abdomen, and the lumbar region, and it has to dilate. And then it kind of narrows down again, and superior to number 1, that white duct, that assumes that that is part of the thoracic duct. So superior to that cisterna chyli, that duct assumes the term thoracic duct. Travels all the way through the chest and then to the left subclavian vein. 3 is pointing to lymphatic, vessels and nodules here in orange. Okay, so 1 is pointing to the cisterna chyli. Its function is to collect lymph from the lower limbs, and legs, and from the lumbar region. You had the pedic portal vein. Oh, and he's asking you to identify the mesenteric vein. It's actually, I think the pointer, well, there are mesenteric veins there, but if you pointed to the white vessel, which is actually at the tip of the arrow, then I would say that's a lymphatic vessel. If I was pointing to the orange, that would be a lymph node. A mesenteric vein is not part of the anatomy that you're required to know. So vessels, I would say lymphatic vessels, lymphatic capillaries, lymph nodes, cisterna chyli, right and left lymphatic ducts. That's it. Okay, this is pointing to red bone marrow. Now, this is one of the joint models you actually looked at in A&P1. Because we were looking at synovial joints, when you had the menisci of the knees and the grayish color, the blue is the hindling cartilage tissue that forms the articular ends of bones. We had tendons and ligaments, and in white you had the synovial membrane, synovial fluid. Same model, but pointing to the red, just representing red bone marrow. So identify, that tissue, red bone marrow. Identify the function, produce all blood cells. I would still use that. Identify the cell that becomes immunocompetent or mature here. Which lymphocyte matures here? A B lymphocyte. It's a variety of different questions. Red bone marrow. It acts as a primary lymph organ. Okay. Um, and its function is to produce all blood cells. And in this case, which lymphocyte matures here? The B lymphocyte. Maturation and immunocompetence really mean the same thing. Okay, and then here we have another naked baby on a plate. This is a similar model. It just has different tone to the skin. Thymus gland produces thymosin, the hormone. It stimulates T cell maturation or T lymphocyte maturation or immunocompetence. And that immunocompetence occurs in the thymus gland. Okay, and then we had the half head model that I was referring to before. I had a big F because there is a big F on that that organ. You have the nasal cavity and sinuses at 2, and then 1 is your adenoid. Right, so we have tonsils. 1 tonsil, the function remove and trap pathogens from the respiratory tract, and you have a mucous membrane. All right, so let me just go back and let me explain what is trying to be identified here. All right, 1 is your adenoid, it's a tonsil. It initiates the immune response due to stimulation of either things that are breathed in or eaten or ingested. But all along the respiratory entrance, in the nasal cavity as well as up here, in the sinuses, we have that stratified pseudostratified ciliated columnar epithelial tissue. We have mucus being secreted there, and remember we said like in AMP1 that sinus cavities warm and moisten air, and they secrete mucus, to capture pathogens. Well, this is their function, so this is where it kind of comes back again now as far as a first line of defense, and secreting a mucus surface to this tissue to inhibit microbial invasion. I don't think I would ask you that. I might ask you a tonsil, but I really wait till we get to the respiratory system so you have a better idea of what that nasal cavity and your sinus cavities. Okay, then we have a spleen, number 1. That's a spleen. The spleen functions to filter blood, right? To me, I'm not going to ask you to distinguish between a white pulp and a red pulp, just that, really throughout the whole thing. And the red pulp isn't just for working on the red blood cells, but it is where you'll find a lot of the macrophages there that do break down those red blood cells. That we have, 3 types of cells there: red blood, T lymphocytes, B lymphocytes, and macrophages. Monocytes and macrophages. The spleen has multiple functions. It's not like, it stores a significant amount of blood in case you have, you start to hemorrhage. It can push out an extraordinary amount of blood to restore the blood volume and therefore blood pressure. But its function within the immune system, primarily one I want you to know, is that it filters the blood, and therefore it has T lymphocytes, B lymphocytes, and macrophages to monitor, the presence of microbes. And yes, it does. It breaks down red blood cells. The macrophages do. It converts that hemi group into unconjugated bilirubin. And here we're looking at a lymph node that happens to be the cortex region, and that is the germinal center of an area. But I'm not going to ask you about the germinal center. So I would just ask you to identify that organ, a lymph node. Is this, what region is this? Cortex, What region is this? Medulla. I'm not going to ask you about the nodules. But here we have a model of skin. This is one of those the smaller ones from the AMP1 lab. All right, so what are we trying to identify here? First line of defense in the immune system, the stratified nature of the epidermis. We have sweat glands, we have oil glands. We have dendritic cells thrown in here in the layers of your stratified squamous epithelium. Okay, skin, first line of defense, protection from bacteria, abrasion, etc. And I thought, so what I would do is be prepared. Let me just go back into this real quick. Is that all? Yeah, that's it. What I would do is be prepared. All right. Let me quick change back to blood. And I think it's this is quite a number, good number. Oopsie doodle of pictures here. Okay. Where the white blood cell pictures might come in. Yeah, I think it's all before this. And so, well, that's a platelet. Not exactly part of the immune system. But here we have a monocyte. So that's a picture of the histological slide of a monocyte. And then here is, oops, we need to go back a couple. Hold on. I'm going backwards through this, not forwards. So hold on just a second. Okay. This is a lymphocyte. If this was a B lymphocyte or a T lymphocyte, what part of the immune system? Is it non-specific or specific, or is it innate or acquired? Well, it's the specific that we acquire through specificity. If that was a T lymphocyte, what is its function? If this was a B lymphocyte, what is its function? Do you see where I'm going with that? So these pictures can come back. Let me back up some more. Hold on. I should have just maybe gone to 1 or 2. Okay, here's the picture of the monocyte. Okay. Identify its function, phagocytosis. Is this part of the specific or non-specific immune system? It's non-specific or innate. Remember, most of these are, really just go back to the beginning of this because this is making me crazy going backwards through all these. But remember that most of these questions are going to be multiple choice or true and false. Occasionally, does it fill in the blank? Here we have a neutrophil. What is its function? Phagocytosis. Does it function in the what branch of the immune system? Does it function in? Or what is does it participate or is it part of the innate or acquired immune system? Here's an eosinophil. That would be the non-specific immune system. But the ones we mostly show you and try to point to is either a neutrophil or a monocyte, or here our friend the basophil, because the basophil helps increase inflammation. Helps increase inflammation with histamine and heparin. So those are the more common ones. Okay, so I hope I didn't confuse you. My bad, for going back and forth, but I find that those models often pop up in the quiz and then possibly in exam 1. And if we're doing this face to face, I actually run into the back and grab those models and bring them back out again, because they'll be scattered around for an exam, and this way you won't be surprised. So again, some of the models from the first lab of blood, some of those are going to be instrumental, in this fourth lab. Okay, that's the end of the lab.